All right. Thank you everyone for joining us today. So we are from the Allen Institute, a nonprofit biological sciences research institute located in Seattle. And our core focus research areas are neuroscience, cell biology, and immunology. So we're going to hear from Jacqueline Bendrick, who is a research associate in the Allen Institute for Brain Science, to get us started. I'm going to start with some questions for Jacqueline to just get you started hearing about her career and think about things that you might want to ask, and then we'll have time for questions from you. So Jacqueline, to get us started, can you tell us about your science journey from high school to now?
Sure. So in high school, I was fortunate to have two really great science teachers, both in biology and chemistry. And so I knew I wanted to go into college in some sort of scientific field, but I wasn't initially sure which one I wanted to choose. So I applied to a lot of scholarships, which it sounds like some of you are as well. And I received a small chemistry scholarship at the university that I ended up going to, which is Furman University in South Carolina. So I decided, hey, I got some money offered, let's do chemistry. And so I went in and I explored the major a little bit more. And found that they had a biochemistry specific path that I could take. And so part of that path had me take a genetics class in the biology department. And the professor was amazing. We got to do this really fun lab where we got cheek swabs donated from dog owners on campus. And then we had to determine using molecular assays what type of dog we had, like what breed. And so at that point, I realized that what I really loved about science was a little bit bigger than what chemistry had to offer. Single molecules are great, but I really liked how they worked in systems. So I switched my major to biology, and I joined a research lab studying how skin cancer cells spread throughout the body, and just had a wonderful time in that lab, published some work detailing how cells talk to each other and how they coordinate moving throughout the body. So sort of simulating in a dish how cancer metastasizes in the body. So I got a paper out of that and applied for jobs. I know long-term I want to go and get my PhD, but I wanted to get a little bit more experience in other fields of biology since there's so many different interesting areas to explore. So I applied as far and wide as possible, which I highly recommend when all of you are out in the workforce. You never really know what's meant to be. And I ended up here at the Allen Institute for Brain Science. I had zero knowledge of neuroscience, and I've been learning on the job every day, and I just love it.
So can you tell us about an average day, both now and pre-pandemic, in your job as a research associate?
Sure. So for a little bit of context, I'm on the genetic tools team here at the Allen Institute. And so a large part of my job is designing, building, and testing these viral genetic tools. So working with viruses might sound scary, but these are, they're called adeno-associated viruses, and they're basically harmless. They already infect the vast majority of the human population and have no adverse effects. So the great part about our work is it's a good field to, our tools could be used for gene therapy down the road. So being able to actually make little changes in people's DNA to fix disease. So with that context, an average day, it sort of depends where we are in what we call a pipeline. And basically a biological pipeline is, we have this long process that we move through, and then we take big batches through each step in the process. So currently I've been spending most days doing the very final step in the process, which is testing our tools and seeing if they work well. And so I'm in the lab pretty much all day and doing a lot of staining. So basically adding a bunch of different chemicals to slices of brain from mouse to test and see. That's my background actually. This is staining for RNA. And then looking to see if the cells that we've made glow brightly with our viral tools are the ones that we were hoping to. So pre-pandemic and now are pretty similar. In the beginning of the pandemic, I transferred to working from home for a couple months and was doing more data analysis, making figures for papers, looking through the mouse genome, trying to pick out like what's going to be our next tool to build. So it's a fun job.
And how long have you been at the Institute?
I've been here over a year and a half.
So what is your favorite thing about your job?
I love how every day I get to come in and learn something new, and especially how I get to learn things that no one else in the world knows. And I think that's a really unique part about science, in that every day you're discovering something new and inventing a new process. And I love not only learning new skills, like new techniques in the lab, but also developing greater knowledge of the brain and how these different parts of the brain work together, and all the different types of cells that are in the different parts of the brain. So I think that's a really unique part about science.
So much as we give our best efforts to all of our science, sometimes it doesn't work the way we planned. Can you tell us about your best science failure? Whatever that means to you, best learning experience, funniest, most surprising.
Yeah, so actually I'd say my best science failure was in undergraduate. So I had spent a semester reading papers and like learning lab techniques before I started this like 10-week internship. And so I felt like I was so ready to go. And, when you do labs in classes, everything is so pre-planned and you basically know it's going to work because, people have done it 100 times before and it's like perfectly designed. And so I got to the lab my first week, and this is a tissue culture lab. So what that means is we were growing human skin cancer cells in dishes. And that takes a lot of time to prepare for experiments because you can't just like pull stuff out of the fridge and start going. You have to wait for the cells to grow. So I spent 5 days waiting for my cells to be like the perfect stage for doing this experiment. And the experiment involved scratching the surface of the cells, so they're all on the bottom of the plate, and then measuring how fast they migrated back into this scratch that we made. And the first time I did it, I scratched way too hard because I'd never done it before. And the entire sheet of cells just came up off the dish and floated away. And so that was basically, we just had to scrap the whole thing and start over. And I think that was a great learning experience though, because not only did it make me appreciate that behind every paper that you read or every experiment that you see that's successful, there's many, many that failed. But it also really made me reevaluate, that not everything that seems like a failure actually is. And that for every lab experiment that doesn't go as planned, there's always a little golden nugget of information that you can pull out that becomes really valuable in the future.
You raised a great point just there about the difference between studying science in a class and learning it from a book, or running labs that are pre-planned and sort of have a known result, versus working in a research lab. Can you expand on that?
Sure. So I think in school, I enjoyed doing labs, but I have to say it was always sort of something that I was like, oh, I got to go to a 3-hour lab now. Like, people who are other kinds of majors don't have to do this. But actually working in a lab all the time, I think is so much more fun. And part of that, I think, is you have a little bit more creative freedom in that not everything is scripted. And also the results are much more meaningful. So I think, coming in every day, I'm excited to see, what the results of, maybe many weeks of labor have been. And I think the fun thing about being in a real lab too is that you get to learn not only how to do the techniques, but a lot of the backstory in, how those techniques were developed, or, fun, cool ways that you can do things slightly differently that might be easier to get a better result. So it keeps you on your toes more so than academic learning. But they're both very important. You've got to have the background before you can transfer into, real world applications.
Yeah, a lot of things that don't get captured in textbooks or journal articles that you really learn from doing it in a lab. Like, little methodological quirks and tips for how to analyze data, not just putting it into the context of existing knowledge.
Definitely. Yeah, even little things like someone taught me, oh, when you put like a fluid in the tube, just like turn the lid to the left. So then, you've already done that one, because, sometimes we'll be doing these massive rows of things to pipette. And then it's like, oh, it's a foolproof way. You never have to remember like, oh, am I on the 4th one or the 5th one? It's like fun stuff like that. It's nice to be in a community too in a lab.
So what do you wish you had known when you were in high school about what a career is like in or around science?
It's a great question. I think I wish I had known that having as much experience, whether internships or taking classes with labs, I wish I knew how important that was for getting a job in the real world. I think, it's easy to have a lot of things going on in college and high school, and to, sort of make other things the priority. And I realize now how much easier it is to get a job, or to get, a degree, when you have a little bit more real world experience. So I think I wish I would have started earlier, but I think everyone says that about something in life. So just getting as much real world practice as possible.
Can you tell us about your first internship? How did you find it? Why were you interested in it? What was the experience like during the internship?
Yes. So it was my junior year of college. So I played golf in college to finance my education. And so I really didn't have a lot of time. I felt like to actually do research, but we were required to do, a real world research experience before we could graduate, and to write a thesis on that. So I started exploring the different labs at the university, and they had a showcase where you could go and kind of talk to the professors and hear about their research. And so I watched the presentation for this one professor, and he studied skin cancer. And that's cancer is something that's very relevant to me. Both my grandmothers passed away when I was very young due to cancer. And so it has a personal connection. And I thought it would be fantastic to be able to work in a field that could, one day help treat different types of cancers. So I applied for the job. And so the professor had a bit of a unique application process, in that he didn't care how much experience you had coming in. He cared how much passion you had for actually engaging in science. So I read papers, and then I'd come back to him with questions. And so then we applied for grants, because in science you can't do anything without money. So got to learn about that process as well, and was fortunate enough to receive enough funding to do a summer internship. And I spent 10 weeks that summer studying these skin cancer cells and how they talk to each other and coordinate migration. And then I decided to come back the next summer because I enjoyed it so much. So I did 2 summers in that lab, and also worked a little bit during the year as I could around a golf schedule, and was fortunate enough to have enough data to publish a paper. So that was a really fun process as well, getting to see the science from the beginning of the story all the way to the end.
So at the Allen Institute, our research is all basic science, and it sounds like this lab was as well. Can you talk about the difference between basic and translational research and what that means to you?
Sure. So I think basic science, a lot of people interpret this term incorrectly. So it's not sort of like this boring, like, oh, these people just like go do random stuff that's not relevant. Basic science is really the foundation on which all treatment for disease or medication has to rely. So in basic science, we're really seeking to answer these very, small but important questions. So how do cells talk to each other? What chemicals are involved in different processes? How do cells move? How do neurons talk to each other? How do I move my hand? Like, we have such a limited understanding of how a lot of these processes work, especially in neuroscience. There's just so much left to discover. And every day, you read a new paper, it's just groundbreaking about how, this one very small cell type in the brain is what allows us to do something amazing. So whereas translational science takes this big foundation, which is like the Allen Institute, all of our work is like building the bottom of the pyramid that allows us to climb up to treat disease. And translational work takes that information and uses it to create drugs or to create treatments for disease, and translates that into a context on a larger scale.
If you had unlimited resources, as you mentioned, science runs on grants. If you had unlimited resources, what research question, idea, or project would you like to spend your time exploring?
Oh, wow. That's a good one. So many interesting things. Well, I would say I would probably work on understanding more about how I've always had a passion for the pancreas, which seems kind of random, but I have type 1 diabetes. And so I grew up attending these research symposiums. And I would love to understand how the immune system interacts with these insulin producing cells in the pancreas. And there's a lot of people doing good work on that, but with unlimited funding, I feel like I could know everything I wanted to about how that system works and how to better treat autoimmune diseases overall.
So grants are, I think, one part of this. What do you wish more people understood about your research in neuroscience, the field of neuroscience in general, the world of science as a whole? I think grants might be one thing that's how science gets done, but what else?
Yeah, that's a great question. I think if I were to have like one big thing I wanted everyone to know about science is how important laying this groundwork is. And I even talked to like my grandpa about what I do. And he's sort of like, why would you study mice? Like, how does that translate over to people? And I'm like, well, we can't do this stuff on people. There's a lot less availability of subjects. But there's so much that we can learn from this large body of kind of smaller nitty-gritty research, and how important it is to have that foundation to understand, how drugs are going to work in the body or how we can stop cancer from spreading. And I do think that foundational or basic science research is undervalued in the general community. I think in neuroscience as well, people seem to think that, because we have drugs that can treat depression or different conditions in the brain, that we know everything. And it's really not true at all. There's so much that we learn every day, in universities around the world. And I think with the brain, it's really important to fund this research still, and that treatments for so many different types of diseases could come a much further way if we had a better understanding of how things work at this small scale.
We have a question from the audience. I'm wondering if Jacqueline has encountered bias in her education or work, gender, age, racial, etc., and how she managed it.
That's a great question. I'll say overall, I've been really fortunate to not have encountered significant bias in my work, but I think part of that was due to the fact I really targeted my attention to working under professors or principal investigators, or PIs as they're called, that were really diverse people and that were very accepting. So I think it's important to kind of feel out the space you're heading into before you go there. And I've definitely encountered people who have, gone to labs and really hated the professor because, they felt like that they weren't valued because, they were a woman, or, various other, a different ethnicity. So I would say, just keep an eye out and trust your gut. And when you meet someone that you really feel like you click with, that's a good place to go work. My manager here, I talked to her once and I was like, this is someone I want to work with. Like, she has a positive team. She really nurtures a great working environment. So that would be my biggest piece of advice, to avoid as much as possible the negative parts of the workplace.
So PhD programs are not like a bachelor's or master's program where you spend most of your time in class. In a PhD program, you spend most of your time working in a lab. And the best advice I've ever heard for figuring out what lab to join in a PhD program was choose a mentor, choose somebody that is supportive for exactly the reasons you describe. For working in a lab, for joining a lab for a PhD, for working in a lab as a professional in science, choose somebody that you work with well, who is supportive and accepting.
Yeah, it's great advice.
So changing gears a little bit, what do you like to do outside of work? You don't spend 24/7 on science.
That is true. I love to play golf. That's my passion. So I play with a golf group in Seattle, a bunch of women who are mostly older than me, but it's fun to talk to them about work. And I actually ran into one of them is a rep for one of the companies, 10x Genomics, that we use for all of our research. So it's fun to get out and do something different during the day. But
and you played competitively,
science.
Is that correct?
Yes. Yeah, I played Division I level in college.
So another question from the audience: what kinds of interdisciplinary or crossover skills are necessary to be successful and happy as a research associate in neuroscience?
That's a great question. I would say, so I always heard from people who had left college or graduated before me, like, I didn't use anything I learned there. Like, I don't know what this degree was about. And I found the exact opposite. Having good writing skills, having good communication skills, being able to work on a team, having a good planning, being able to coordinate. Sometimes I have like 10 or 15 experiments going at the same time with, different time points. So I'd say it's, really important to be organized and make sure you know that you have a system that works for you as you try to scale your work up. And obviously the science background is really important as well. Having a basic knowledge of math is important. Depending on how far you go in neuroscience, calculus can be a really, key part of your research as well. So I think that neuroscience really crosses many, many disciplines, and also understanding some of the history behind what you're doing and why we do things the way we do, particularly, for different assays, or it even helps to just appreciate, like one of the RAs in my group had to used to do this assay called polymerase chain reaction by hand when she first started, and would have to like go from the ice bucket to the warm bucket to the ice bucket, and now we have a machine that does it. So yeah, I think that it really crosses many, many fields.
And that is the same PCR test that you might have heard about in the news lately from COVID tests.
Yes.
Another question from the audience has come in. How do you keep yourself updated on what's happening in the field of neuroscience?
Yeah, it's a great question. A lot of it is reading papers. I get a lot of those from colleagues. So my team has a channel on Teams where we post things that we think other people might find interesting. And everyone in my group has sort of a different specialty. So my manager has a great background in genomics and epigenetics, whereas another member of my team is more of a computer scientist, and so she has really interesting posts about machine learning and quantifying various assays that we work on. And some of it's just wandering around the literature. You can go on major journals, just look at the webpage and see, okay, what's been published recently. And there's some neuroscience specific journals and some more general ones as well. But it's always interesting to see what people are coming out with. There's so much amazing science coming out every day.
We do have time for a couple more questions. So if anybody has a question for Jacqueline, please go ahead and pop that in. And in the meantime, can you talk about, so you said you plan to go to grad school at some point. What do you anticipate your grad school experience or goals will be?
Yeah, so I'm actually planning on applying this year to grad school. And I'm looking at programs with a neuroscience kind of umbrella, but looking at labs that particularly focus on cell and molecular biology. And so what that means is I basically get to do what I did in undergrad and what I do now in one program. So I'm really interested in how individual neurons in the brain, how they talk to each other, both from firing electricity and also from letting out these cell signaling molecules. And also how their DNA is modulated, so how it's changed between cell to cell to create very, very different looking and acting neurons. So I think that's a really cool part of the brain, or actually the whole body, in that every single cell in your body has the exact same key. It has the same DNA, but yet your eyes look very different from your hands. And the same thing is true in your brain. Different neurons in the brain do very different things. And I would love to study more about how that happens and how we can use that to treat disease as well.
So we'll give another moment for any additional questions. Oh, looks like we've just got one. Oh, never mind. And we'll give another moment for any final questions. But while those might trickle in, do you have any overall thoughts that you want to share with students about a career in neuroscience working in the lab?
Yeah, I would say overall, just dream big and don't accept no. I think a lot of the like this job here and the research I did in undergrad, I didn't know if I was going to get in, and it was sort of felt like a stretch goal. But if you're just persistent and you try hard and you just talk to people, reach out to, the heads of labs that you're interested in working in, or, reach out to professors in the program that you're interested in applying to, and just make that personal connection. And it really goes a long way. And don't feel like you're underqualified. You never really know what people are looking for, either in a job or an internship. Sometimes you might have a skill that isn't on the job description that the professor is really interested in having in their lab. So dream big and see where it takes you.
Yeah, as you mentioned, neuroscience is such a huge field. It's not just that cellular and molecular or viral or like benchtop wet science. There's a whole world of related work as well.
Mhm. Definitely. Yeah, we have a member of our group who doesn't do any wet lab science. She just does computer programming. And the work she does is amazing. She takes like the image I have in my background here, and she can spit out which cells are labeled what color and how many dots there are per cell, and work that manually would take months to do. So there's really diverse careers in science. Just explore what you can and see where you end up.
All right. Well, oh, we have one more question. How hard is it to get a paper published? And can you talk in general about what the process of getting a scientific journal article published is like?
Yes, that's a really good question. So I'll start with the process because that kind of relates to how hard it is. So the process is you usually spend at least, I'd say minimum 6 months to a year, sometimes more, collecting data, depending on how many successful experiments you have. And you build upon the data you collect and kind of expand in more directions. And then once you feel like you have enough, you sit down and you say, okay, let's get this organized. Let's see, what we have for a paper. So it's a lot of outlining, a lot of collaborating with colleagues who have helped you with the work. And then usually you start with putting together figures, so taking images and graphs, and then sitting down and exploring relevant literature. So that's a big part of paper writing, is reading lots and lots of papers that are in the field, and then you cite those in your paper. So you have to connect the work you've done with what other people have done, and really emphasize how what you've done is different than what others have done in the field, and how it's actually significant enough to be worthy of being published. And then you submit it to a journal and you cross your fingers and you hope. And then they either say, nope, we don't like it, sorry. Or they say, yeah, it's pretty good, but you need to make these changes before we'll accept it. And they send it out to independent reviewers who will submit that feedback. And then very rarely, basically never, they'll be like, yeah, it's good as is. And that never really happens. And the difficulty of that process depends on how basically high of a tier the journal is. So just like, different colleges and universities have different reputations, it's the same with journals. Getting into some of the top ones like Nature is, very, very challenging, whereas getting into some lower journals is still a process, but it can be much easier.
And you mentioned that your colleague, who does a lot of the data analysis for your team, made that beautiful image that you have as your background. Can
Yes.
you tell us what that image is?
Of course, yeah. So each dot you see in the image is a piece of RNA, which you may have heard of in one of your classes, but it's like the messenger that goes between the DNA and the cell and then comes out into the cytoplasm to make a protein. And so this is from an assay that labels single molecules with fluorescent, which are basically just glowing colors. So this is a piece of the mouse brain, and the red label and the pink label are excitatory and inhibitory neurons, which are sort of these two classes that are mutually exclusive. And then this is to validate a new mouse line that we made. So the green is the RNA from one of the fluorophores in our mouse line. But it's really fun to image these. And this is just a small snippet. We usually do like whole sections worth.
And what is a fluorophore?
Yes, it's like a glowing protein. So there's all sorts of different types, and they go in all sorts of different colors. But when you shine a laser on it of a certain wavelength of light, it emits a different color. And so then you can see it on these special microscopes.
Great. Well, I think it is just about time for us to transition over. So thank you so much, Jacqueline.
Thanks for having me.